All right, class. So in this last video on chapter 10, we'll wrap up our last few concepts. So in the last video, we talked about types of contractions and tension in a muscle. And so in this video, we're going to start with looking at how muscles fuel those contractions. So how they produce the energy, the ATP that they need in order to do all that movement. And then we will wrap up with a quick look at the structure and function of cardiac and smooth muscle tissue. So starting off with energy. Muscles require a lot of energy. And this may be the first time where we actually had the opportunity to do some math to figure out the number as far as the total amount of ATP molecules needed to fuel a particular type of work. You're probably familiar with the idea now that one glucose molecule can result in a total of 30 to 40 ATP molecules if it were to go through the entire process of aerobic respiration, right? So one glucose gives us 40 ATP at the high end. So now if we break that down and try to think about how much energy a single muscle is utilizing per second, this stuff starts to get a bit mind-blowing. So I guess I'm skipping to the bottom here. One small muscle is composed of thousands of muscle cells. So one small muscle organ, like the muscles of your eyes, right? Thousands of muscle cells in that teeny tiny muscle. Each muscle cell contains on average around 15 billion thick filaments. One thick filament requires 2,500 ATP molecules per second to power those power strokes, release power stroke. Right? So per second, one thick filament using 2,500 ATP. We can multiply that number by 15 billion for the number of thick filaments in each muscle cell, and then multiply that number by, let's say, 2,000 to get an idea of the total amount of ATP used in a tiny muscle. That's an insanely large number to consider. Right? So muscles are using a lot of energy. And they try to prepare for this. They have some special structures as well as their own stored energy sources that some of your other tissues don't have. So muscle cells have some ATP stored and ready to go at a moment's notice. But that's only enough ATP to fuel on average around 2 seconds of a contraction. And so then we need some way to immediately replace that ATP to help assist with that maintained contraction so that that gives the cell enough time to start breaking down these other sources of ATP like fatty acids and glucose. And so during that time, what's fueling the muscle contraction is this other molecule called creatine phosphate. Creatine is a short polypeptide, so a very small protein that is carrying a single phosphate group. And its job is to just give up that phosphate group to ADP. So you use ATP, it hydrolyzes into ADP, just two phosphates, and loses that third. Creatine phosphate can then donate that third phosphate right back to it. So now we have another ATP molecule ready to go. And so creatine phosphate, there's enough of that stored in a muscle cell to fuel around 15 seconds of work. That's a much easier molecule to store and keep ready. So then after those first 17 seconds of work, your muscles are then heavily relying on cellular respiration to make up for these ATP needs. And so muscles have stored glucose in the form of glycogen, which is unique to most of your other tissues. Really, glycogen is only stored in muscles and the liver. So it has its own storage, doesn't need to rely on the liver to put more glucose into the blood. So it is going to break glucose off of that glycogen. It's taking glucose in from the blood. And there's also a lot of fat tissue around our muscles so that we can break down those triglycerides into their fatty acids and use those fatty acids to make more ATP. And another big help with all this is that unique protein myoglobin, right? Similar to hemoglobin in our red blood cells that carries oxygen to our cells and carries carbon dioxide away, myoglobin in your muscle cells holds oxygen and stores it there for respiration. So we're going to look at what's going on energy-wise in your muscles at rest, during moderate activity, and during intense activity, because we are shifting through a few different systems during that. So at rest, okay, your muscles are essentially building up their storage so that they are ready for when they need to do more intense activity. So they're mainly relying on fatty acids as their energy source at rest. Fatty acids contain almost or more than double the amount of calories per gram than carbohydrates do. So as long as there is time to do the multiple chemical reactions needed to break down a fatty acid, and we have oxygen available, because fatty acids can only be used for aerobic respiration, our muscles are going to rely on that. So at rest, your muscles are mainly using fat as their energy source, and they are absorbing glucose from the blood in order to store it as glycogen. So we're building up our glycogen stores, we're maintaining our cells with fat, and then we are storing extra ATP and creating extra creatine phosphate, again, with the goal of being ready for when we need that energy. So then we start getting into some moderate activity. And this is where those steps that I was talking about in the previous slide come into play. Right, so we have ATP to fuel the first couple seconds, creatine phosphate to help back that up and replace it while respiration is essentially continuing. So we're still going to be able to tap into fatty acids for some energy as long as we have oxygen available. But more and more, we're going to start digging into that glycogen storage, pulling out glucose and fully breaking glucose down to help with that ATP production. So we have a mix of both depending on the intensity. The more intense the activity is getting, the more we're going to shift towards glucose and away from fatty acids because oxygen is going to become less and less available. So if we get into super intense activity, where essentially your muscles are utilizing oxygen and ATP faster than oxygen can get to your cells from your blood, in this state, your muscles are purely relying on glucose as their energy source. And very few of those glucose molecules are going to be able to get into the mitochondria for those later stages of respiration. And a lot of those glucose molecules are being broken down just through anaerobic respiration. So we do glycolysis, splitting that glucose in half, getting a couple ATP molecules as a result. But then we still need to do something with this pyruvate, or really what needs to happen is the electron carriers need to be recycled. And they get recycled by interacting with pyruvate, which turns pyruvate into lactate. And anaerobic respiration is also going to lead to the buildup of these protons. And so lactate then needs to leave the muscle cell and go to the liver to be recycled, which is an extra energy need and slows down your metabolism in your liver. And this buildup of protons is going to increase the acidity, right? Decrease the pH of your muscles, which is going to inhibit your sarcomeres from being able to actually contract. So that's when we start to feel that fatigue, that sluggishness in our muscles is due to the buildup of these protons from anaerobic respiration, thus increasing the acidity and slowing down those muscle contractions. They are less able to actually contract. Okay. So that's how our muscles fuel ourselves. And some of our muscles are better at this job than others. We have this split between fast twitch and slow twitch muscle fibers, and there's even an intermediate muscle fiber. Or you may be more familiar with this with respect to meat that we consume, red meat versus white meat. That is the physical appearance of this fast versus slow twitch muscle fiber. So slow twitch muscle fibers, like what we see shown here from the gastrocnemius muscle, tend to have a lower concentration of like actual muscle, of myofibrils and sarcomeres compared to a fast twitch muscle fiber, but they have a lot more supportive mechanisms and structures in there. So a slow twitch muscle fiber would have a lot more fatty tissue around it to provide those fatty acids for sustained energy. They have a lot more glycolytic enzymes for the breakdown and for the storage and breakdown of glycogen, again, for that sustained energy production. And they have a lot more mitochondria for aerobic respiration. So although there is a lower density of the actual contractile units, all these other supportive structures allow these slow twitch muscle fibers to continuously produce tension over a long period of time, right? They're better for endurance. So this would be a red meat. Right? It would show up as red. There's a lot more myoglobin, right? A lot more blood support as well, so a lot more capillaries within these slow twitch muscle fibers, and so they have that reddish color like blood does. Fast twitch muscle fibers don't have as nearly as much of that support. It's mostly muscle. It's there to produce a quick tension and react fast, but it is not structured for sustained contraction. Okay? So these are a lot of our smaller muscles, like the muscles of our eyes, like our lateral rectus muscle, what's shown here. So these tend to show up as paler in color since they don't have that capillary support. They have less myoglobin, less mitochondria, less glycolytic enzymes. It's mostly just muscle tissue. And so this produces our white meat. Okay, so if you think about like a chicken, they have the white meat of their breast muscles and the dark meats, the red meats of their leg muscles. Because chickens have evolved to not rely on flight as a sustained form of motion, they can flap their wings for a brief moment of time to fly up into a tree to escape danger, but they don't fly over long distances or migrate. So the breast meat of a chicken is made up of these fast twitch fibers, and they look white. They have a lot less of those capillaries, those red meats. Whereas their leg muscles are more like these slow twitch fibers, that red meat, that dark meat, because they are relying on their legs a lot more. Or if you compare that to the breast meat of a duck, which does fly for long periods of time, they do these long migrations. So their breast meat is darker, it is red meat. Right? And you can train your muscles to shift more towards the characteristics of a slow twitch fiber or more towards the characteristics of a fast twitch fiber. Right? Your bodies are very malleable in how they perform, and so they will shift based on training. So if you train for endurance, some of your fast twitch fibers will start to shift in their characteristics to perform more like an intermediate or slow twitch fiber, right? They'll start generate more mitochondria. There will be more capillary structure and support going into them. Okay, so you can shift these things through training, which is a pretty cool thing about our muscles. All right. And so our very last concept here is just taking a quick look back at kind of what we already addressed early on in chapter 4 about the different types of muscle tissue. So just reminding you the differences with cardiac muscle and smooth muscle structure compared to skeletal muscles, and then applying some of these ideas that we've learned about how contraction works. So cardiac muscle is similar in function to skeletal muscle. We still have sarcomeres, and that's going to create these striations in the muscle. But rather than having these really long muscle cells that go end to end with multiple nuclei, so producing one contraction at a time, filling the full muscle with that single contraction, cardiac muscle is structured in a way that we can get this pacemaker, so kind of this domino effect or wave effect, where a cell contracts, then the next cell contracts, and the next cell contracts. And that's very important for how our heart functions, right? Pushing blood from one chamber to the next. So it would not be helpful, or it would not function as well if the entire heart pumped at once. Instead, we produce this wave which directs blood flow throughout your entire body, right? So that comes from single cells next to each other, separated by these intercalated discs. Let's get back here. So again, we have striations, we have sarcomeres, similar function. We got mitochondria in here to help with powering, but each cell is just a single cell, does not go the entire length of the organ. Okay? So a single nuclei. And then the next cell, the neighboring cell, is attached to it by these strong intercellular connections, creating these intercalated discs. Right? What we see with this squiggly line here, or this separated intercalated disc here. And so this allows for this cell to contract separately from the neighboring cell, which is then going to stimulate a contraction in the next cell, again, producing that wave to help push blood, again, through the chambers of your heart. So most of the actual function is the same, just a bit of a different structure, giving a slight deviation in how the organ functions. And instead of all of these parallel long muscle cells contracting at the same time, we have separated muscle cells so that we can produce that wave. Smooth muscle, however, gets really funky looking. So rather than pulling in this linear way that we were thinking about with skeletal muscles. We still have thick and thin filaments. We have myosin and actin that are going to pull on each other, but they are kind of distributed unevenly throughout the smooth muscle. And those thin filaments, instead of being attached to titin, which connects them to that line where all of the sarcomeres are kind of like attached or anchored to the same structure, the thin filaments here are anchored to a different protein called desmin, or sorry, these dense bodies. And those dense bodies are attached to intermediate filaments called desmin. So the main method of contraction is still the same. It's myosin heads pulling on actin. But instead of that creating this sliding filament model, that actin is going to pull on these dense bodies acting as anchors. And that's going to bring those dense bodies essentially closer to the center of that organ. And as those dense bodies move, they are going to bring with them this long, thick network of intermediate filaments. And so altogether, these dense bodies being pulled to the center, like the nucleus of the cell, or of the organ, sorry, and all these intermediate filaments around it being pulled in kind of like a fishnet. If a fishnet was surrounding something and it was all squeezed towards the center, that produces this contracted form of a smooth muscle. So instead of these muscles contracting linearly, they contract more in a three-dimensional kind of from the outside to the center type of thing. So a little bit different. And that's really it. So that is the end of this chapter on muscles. Thank you guys for being here, and I will see you in the next one.